So good evening, everyone. A warm welcome to the 14th Pan India Journal Club session. So let me share the screen and we'll be beginning today's session. So today's Journal Club session is going to be on the topic of the role of dietary fiber in modulating the gut microbiota dysbiosis in diabetic patients. It's a systematic review. So let me first give you the flow of today's session. So the introductory part will be done by Dr. Dharani and Dr. Drishit. Methodology part will be done by Deshna Oswal. Results will be done by Srinidhi and Suleiman. And the discussion and conclusion part will be done by Dr. Preetham and Dr. Sindhura. So I would like to, I would request Dr. Suleiman to please take over the panel introduction part, please.
Thank you, ma'am. Am I audible?
Yes, yes.
Oh, thank you, ma'am. Greetings to everyone present in today's 14th Pan Journal Club. I am Suleiman, a second year MBBS student from Bangalore Medical College and Research Institute. And this is a team which is working to spread awareness on better daily dietary choices. So today I'll be giving a small introduction for the PAN team. Since prevention is always better than cure, which forms the fundamental idea of the PAN organizations, it is a spirited team which is growing towards an approach to build a network globally and nationally to improve the nutrition system with the power of right dietary choices and to prevent diseases. Since it's always noticed that the change is always contradicted, so the PAN team is working rigorously to appeal to the society through evidence-based guidance. And for the same purpose, to reach out nationally and spread this positive movement, Pan India was established in September of 2022. With the chronic diseases, cardiovascular diseases, and diabetes reaching new heights, Pan India as a team has got an aspirational approach. We organize polls, quizzes, feedback forms, query answer sessions, journal clubs, in person CMEs, discussion forums to disseminate this information to a wider group and through the social media platforms such as WhatsApp, Instagram, and LinkedIn. The PAN team is headed by a director, Dr. Rajina Shaheen. It also houses really spirited volunteers, interns, ambassadors, which include a huge spectrum of professionals, right from the medical students yet in medical school to working doctors, nutritionists, and much more, who are all with the goal of improving the present broken nutrition plate. And moreover, especially emphasizing it includes all of you who attend the PAN events and form the backbone of methods to spread our message. And it's quite evident that the team is showing a positive growth. So anyone who's interested in the same goal towards improving your knowledge on dietary choices, we welcome you with open hands to be a part of this movement. With this small introduction, now I would hand over to the introduction of today's journal club paper, which is on the topic of the role of dietary fibers in modulating gut dysbiosis in diabetic patients. Thank you.
Hello everyone, I'm Dr. Dharani. Yeah, we all know that diabetes is one of the most prevalent non-communicable diseases these days, and the numbers are rising to over 642 millions by the year of 2040. And diabetes not only compromises the quality of life, but also has significant impact on mortality and morbidity. And some of the complications that diabetes have is microvascular and macrovascular. Microvascular like retinopathy, nephropathy, neuropathy, and macrovascular like stroke, heart disease, and peripheral vascular diseases. Next slide, please. Yeah. Some of the common risk factors for the development of type 2 diabetes is overweight and obesity, and lifestyle like physical inactivity and genetic predisposition, gut microbiota dysbiosis. So whenever there is a high sugar intake and also low fiber intake, when we consume more of sugar, this can draw water from the cell walls of the bacteria through osmosis, and it can lyse the bacterial cell walls and it causes bacterial dysbiosis. Next slide, please. So here is a study where the number one, it shows thick mucus which is growing and which is healthy. And the second, once we consume Western diet or very highly processed food, the thickness of the mucous membrane is getting decreased, and you can see there is a change in microbiota. And within 1 to 3 days, there is significant reduction in the mucus production in the intestinal humans. And after 5 to 6 weeks, the host develops the compensatory mechanism. And during this time, if we just supplement with the fiber like inulin, which is a very resistant starch, and also bacteria like bifidobacterium, and you can see it will grow back to the initial thick mucus and normal microbiota. Next slide, please. So how do we restore the gut microbiome? So if at all there is a gut microbiome dysbiosis, here are some of the things we can do. We can supplement with prebiotics like complex starch and probiotics and postbiotics. Postbiotics is nothing but the products which are formed from the prebiotics. And also symbiotics, which is a mixture of pre and probiotics. And yeah, what is the benefit of having good microbiome in the gut? So it provides gut immunity, and it revives enterocytes, mucosal integrity, and it balances the pathogens inside the lumen. And what are the negative effects if we have a disrupted gut microbiome? So it can lead to oxidation, stress, apoptosis, and it can be an oncogenic. Yeah, like I've explained in the previous slide, this is the same thing. There are some viruses which can cause viruses and bacteria which can cause malignant transformation. And studies have shown that there are use of dietary interventions, including dietary fiber, has been promoted by researchers and practitioners involved in diabetes care and management. There are different kinds of fiber. Some of them are polysaccharides, resistant starch, and resistant oligosaccharides. And this is the molecular structure depending upon how many carbohydrates that it have. Next slide, please. And these are some of the high fiber foods, and we can include the native varieties like complex carbs like jowar and ragi, and even that contains high amount of fiber and good for diabetics as well. Next slide, please. Yeah. This is a brief introduction regarding the types of fiber, which is soluble and insoluble. Like we know, the soluble fiber just attracts the water and helps in slow digestion so that there is no postprandial sugar spikes, and it also lowers the cholesterol. In insoluble fiber such as roughage, it is made up of cellulose, and it just adds bulk to the food and helps in relieving the constipation. Thank you.
Yes, so good evening, everyone. Thank you for joining this call after a busy day. So how this intervention might work? So, as discussed earlier, gut gut microbiome has a huge potential on how the food is being processed. Normally, human gut bacteria contains 4 main phyla, as shown in the pie chart. Diet plays an important role in modulating the microbiota, and they leads to production of short-chain fatty acids, which are mainly acetic acid, propionic acid, and butyric acid. They are the source of energies and are also involved in metabolism, immune system, and proliferation of cells. So, accumulation of short-chain fatty acids leads to secretion of GIP and glucagon-like peptides, leading to reduced fat accumulation. But what happens is when we eat too much of junk food, less fibrous food, there is a change in this gut microbiota, which leads to dysbiosis, and can lead to insulin resistance in type 2 diabetes mellitus. Poor diabetic habits can lead to dysbiosis, including overgrowth of proteobacteria and reduction of bacteroids, which is the second most prominent bacteria. Next slide, please. So there is some evidence that proliferation of proteobacteria may lead to energy disequilibrium, suppression of growth of other bacteria, and development of diseases, including type 2 diabetes mellitus. The consumption of high fiber diet may alter the gut environment and provide proper growth and proliferation and production of short-chain fatty acids. The main thing about short-chain fatty acids is they are not only source of energies, but they are transported to the peripheral circulation through portal vein to the liver and peripheral tissues. So, not only will poor dietary consumption reduce bacterial diversity, but also reduces short-chain fatty acid production and shift towards the utilization of less favorable substrates such as dietary and endogenous protein sources by the gut microbiota. And the main thing is that the fermentation of these proteins into amino acids by the microorganisms can lead to reduced production of total fatty acids and butyrate, as well as increased production of cytotoxic metabolics that contribute to development of chronic diseases, including type 2 diabetes mellitus. Thank you.
Thank you so much. Good evening, everyone. Am I audible? So let's continue. Why it is important to do this review. Next slide, please. So previous studies show inconsistent evidence or an association between higher intake of dietary fiber and reduced incidence of type 2 diabetes mellitus. Non-digestible oligosaccharides may affect the community of bacteria that make up the gut microflora, and their effect on health is evolving area of research. Other studies have focused on broad subjects of dietary or lifestyle intervention or probiotics and fecal microbial transplantation, and not primarily on dietary fiber. Therefore, no meta-analysis on the effect of dietary fiber on gut microbiota in patients with type 2 diabetes has been conducted. Next slide, please. Therefore, the aim of the study is to evaluate the role of dietary fiber in modulating gut microbiota dysbiosis in patients with type 2 diabetes mellitus. Method of this particular study, this is a systematic review and meta-analysis of randomized controlled trials which relied on PRISMA framework, that is preferred reporting items for systematic reviews and meta-analysis framework. Types of studies included were RCTs, that is randomized controlled trial. Type of interventions was dietary fiber, including a macrobiotic diet. Outcomes measured were relative abundance of gut microbiota, SCFA, that is short chain fatty acid, glycemic parameters like HbA1c, fasting blood glucose, homeostatic model assessment of insulin resistance, that is HOMA-IR, and along with adverse events like total adverse events, diarrhea, bloating, constipation, and abdominal pain. Next slide, please. Search strategies. The search method for identification of studies included electronic searches were conducted using EBSCOhost with links to health science research databases encompassing Academic Search Premier, Medline, Psychology and Behavioral Science Collection, APA PsycInfo, Sinhal Plus with full text, and APA PsycArticles databases. Other databases included Embase and Google Scholar. A reference list of articles were also searched for relevant studies. Searches were conducted from date of commencement of database to 5th August 2020. Search strategy included PICO, that is population, intervention, comparator, and outcome, and use of mesh terms. Searches were conducted independently by 2 researchers and were cross-checked by the 4th. Data retrieved were exported to EndNote to remove the duplicates. Next slide, please. So now data collection and analysis. We'll start with selection of studies, then we'll go with data extraction and management, assessment of risk bias and quality of included studies, finally data analysis and effect size. Next slide. So in this figure we can see, previous slide please. In this figure we can see from Embase, the figures. Next slide. From Embase, there were 90 articles. From EBSCOhost, there were 432 articles. Google Scholar led to 8 articles, and reference list of articles led to 5 articles. Records after duplication, removal of duplication were 471. Then 429 studies were excluded on the basis of abstract and titles, leading us to 42 studies. And then 33 studies were excluded again because of the studies with no outcomes of interest. Review articles, 5 studies with abstract were only 2. So finally leading to 9 articles. So we, in this meta-analysis, we had 9 articles. Next slide, please. Inclusion and exclusion criteria of this particular study was patients with type 2 diabetes, above 18 years of age, dietary fiber as intervention, the gut microbiota, glycemic parameters, short-chain fatty acids, and adverse events as outcome of interest. Exclusion criteria was participants below 18 years of age, patients with type 1 diabetes, and pre-diabetes or gestational diabetes were excluded. Along with that, studies involving animal models and probiotics were also excluded. Next slide. Data extraction and management. Data was extracted by 2 researchers and cross-checked by other 2 researchers. For the meta-analysis data, the author of selected articles were contacted for the original data where possible. Changes from baseline for the intervention were compared with the control in all parameters analyzed. The Engorge digitizer was used to extract data for the genus Bifidobacterium from the graphs. Units of measurements were converted. Median and first to third quartiles were converted to means and standard deviation, respectively. Next slide, please. Assessment of risk bias and quality of included study. The risk of bias for the included studies was assessed using a domain-based assessment tool. The domain evaluated included selection bias, performance bias, detection bias, attrition bias, and reporting bias. Selection bias was assessed using by random sequence generation and allocation concealment. Performance bias was based on blinding of participants and personnel. Detection bias was based on blinding of outcome assessment. Attrition bias, incomplete outcome data, and reporting bias was assessed using selective reporting. And along with this, other bias were also evaluated. The process was carried out using RevMan, that is Review Manager 5.3 software. In addition, to assess the quality of RCT, CASP checklist was also used. CASP is Critical Appraisal Skills Program. Next slide, please. Data analysis. The meta-analysis was carried out using RevMan 5.3 software. In addition, sensitivity analysis was conducted by removing one study at a time from the meta-analysis in order to assess the level of consistency of the results. The assessment of heterogeneity was by means of I-square statistics, and p-level less than 0.10 was taken as the level of statistical significance of heterogeneity. The fixed effect model and mean difference were used for the meta-analysis. However, for short-chain fatty acids data, these were converted into standardized mean difference due to the use of different measurement scale in the outcome of interest, and the random effect model was used for the analysis. Next slide, please. Finally, the effect size. The overall effect of the intervention in relation to statistical significance was based on p-value less than 0.05, and the results of the meta-analysis were presented as forest plots. Thank you so much. Now I would request Srinidhi and Suleiman to take over for the results. Thank you.
Thank you, Deshna. Next slide, ma'am. So now I will be talking about the results and major findings of the systematic review and meta-analysis. 9 studies which adhere to the inclusion criteria for the systematic review and meta-analysis were included. The majority of the studies were randomized controlled trials with a parallel design, ensuring high reliability and validity. One study employed a randomized control crossover design, adding a unique perspective to the data set. Next slide, ma'am. So this graph over here represents the geographical distribution of studies. This analysis included studies from various countries, ensuring a very diverse and comprehensive data set. 3 of these studies were conducted in Italy, and 1 study each from Norway, Japan, Mexico, UK, Canada, and China. So this geographical diversity enhances the generalizability of the study's finding across different population and setting. The tabular column in this slide and the next slide gives the details of the 9 studies included. It provides key information such as sample size, interventional methods, and outcomes measured. So let me give you a summary of the study characteristic. The previous slide, ma'am. So these studies with participants ranging from 55 to 62 years investigated the effects of dietary fiber on gut microbiota, glucose metabolism, and inflammation. Interventions involved either high fiber diet or fiber supplements. The consistent outcomes across these studies demonstrated a very significant improvement in gut microbiota composition and diversity, a reduction in fasting blood glucose and HbA1c levels, and also a decreased inflammatory markers. Next slide, ma'am. Next slide. So next, we will look at the risk of bias in the included studies. First, it is important to note that 100% of the studies showed a low risk of bias in several key areas, like the blinding of participants in personnel. This means that the participants in study personnel were not aware of the group's assignment, preventing bias in treatment and reporting. And the next is incomplete outcome data. All studies adequately addressed any missing data, ensuring that the results are reliable and accurate. And we have selective reporting. There was no evidence that the studies selectively reported outcomes, which means that the reported results reflect all the data collected. However, when we look at specific areas, less than 75% of the studies had a low risk of bias for blinding of outcome assessment. Furthermore, less than 25% of studies had a low risk of bias for random sequence generation. This involves the method used to randomly assign participants to different groups. So many studies did not adequately describe or implement this, leading to potential selection bias. Next is the allocation concealment. This refers to the concealment of group assignments before participants are assigned, preventing selection bias. Again, fewer studies maintain a low risk of bias in this area as well. So despite these issues, it's reassuring to note that all studies demonstrated either a low risk or an unclear risk of bias across all assessment domains, indicating that while some improvements are needed, the overall quality of the study is reasonably high. And then 4 distinct areas of effects of interventions were identified. The effect of dietary fiber on gut microbiota, the role of dietary fiber on short-chain fatty acids, glycemic control, and adverse events. So now I will be dealing with effect of dietary fiber on gut microbiota, and my colleague Dr. Sulaiman will be dealing with the other 3 components. Next slide, ma'am. So according to Zhao et al., dietary fibers significantly increased the levels of acetate-producing bifidobacterium. So acetate is a short-chain fatty acid that benefits gut health. More importantly, this increase in beneficial bacteria also led to a reduction in the production of harmful compounds like your indole and hydrogen sulfide, which can be detrimental to our metabolism. Raymer et al. Found that the control group had a higher abundance of lactobacillus species. Although Faecalibacterium increased in both dietary fiber and the control group, the increase was more significant in the control group with a p-value of 0.038. Interestingly, the dietary fiber group also had a higher level of Coleella bacteria. Despite these changes, there were no significant differences in overall alpha or beta biodiversity. Next is Birkeland et al. Reported a moderate change in the fecal microbiota composition of the dietary fiber group with 1.5% change. The most prominent effects were observed in the operational taxonomic units or the OTUs of Bifidobacterium bacterioids. So this indicates specific targeted changes in the microbiota composition due to dietary fiber intake. Next, Pedersen et al. Found that prebiotic fiber treatment had no significant effect on total bacteria or specific groups like lactobacillus, rosebaria, Clostridium leptum, or Clostridium coccoides. So this highlights the selective impact of dietary fibers on the different bacterial population. Next slide, ma'am. Next slide. So in patients with type 2 diabetes, There was a notable increase in Prevotella cocci, indicating intestinal dysbiosis. However, following a dietary intervention with functional foods, there was a significant improvement in the fecal microbiota compared to the control diet. This intervention promoted alpha diversity and also increased the abundance of specific beneficial bacteria independently of any anti-diabetic drug. Next slide, ma'am. So this meta-analysis involving 2 studies with 80 participants focused on the genus Bifidobacterium. They found a significant increase in the relative abundance of Bifidobacterium in the dietary fiber group compared to the placebo group, with a mean difference of 0.72 and a p-value of less than 0.01. Again, this underscores the beneficial effect of dietary fiber on increasing this important bacterial genus. Next slide, ma'am. So to summarize, dietary fiber interventions in patients with type 2 diabetes led to notable changes in gut microbiota composition. Specifically, an increase in Bifidobacterium species was consistently observed with the highest effect on Bifidobacterium aeruginosus. Positive changes were also noted in bacteriotes and acetate-producing Bifidobacterium, which are beneficial to gut health. However, no significant effects were observed for total bacteria, lactobacillus, Clostridium leptum, or Clostridium coccoides group. So while Collinsella levels were higher in the fiber group, the overall alpha and beta diversity remains unchanged. Now over to Dr. Suleiman.
Thank you so much. I'll be proceeding with the second part of the results, where I'll be speaking on the role of dietary fiber on the short chain fatty acids, glycemic control, and the adverse events. Before we go into the much details of what this meta-analysis and systematic review brings to us, as the colleagues have already spoken about, I'll just give a brief run through of the short chain fatty acids again. So short chain fatty acids mainly are the acetic acid, propionic acid, and butyric acid. These are essential sources of energy which are involved in the host metabolism, immune system regulation, and proliferation of cells. And these also stimulate secretion of hormones, which leads to reduced fat accumulation. So the short chain fatty acids stimulate the hormones such as the gastric inhibitory polypeptide, glucagon-like peptide, and peptide Y. So these short chain fatty acids are also used as sources of energy in the colonocytes, and they are also transported to the peripheral circulation, to the portal vein, and also the peripheral tissues. As we've just understood, next slide please. We've understood that short chain fatty acids have a significant role. So a poor dietary fiber reduces short chain fatty acids, and which inclines it towards utilization of the less favorable substances, such as the dietary and endogenous protein sources by the gut microbiota. Coming in specific to the present systematic review and the meta-analysis which was done, there was a study which shows that the intervention of dietary fiber has a significant increase in the fecal concentration of the short chain fatty acids with a p-value of 0.04, where the p-value of 0.05 less than was considered as significant. And there was a general trend which indicated that acetic acid was positively related to an increased level of prebiotic fiber. And in relation to the meta-analysis, for the total short chain fatty acids, there were 2 studies included in this paper, which total had of 95 participants. And there were 3 studies, each for having 145 participants for acetic acid, propionic acid, and butyric acid. Overall, there was a significant difference of p-value 0.02 between the dietary fiber group and a placebo with a standard mean difference of 0.5. This was pertaining to the short chain fatty acids. We'll move on to glycemic control. So, glycemic control in overall, it reduces the risk of diabetic complications and it enhances the energy and cognitive functions. In poor glycemic control, always increases the risk of complications such as cardiovascular diseases, renal failure, neuropathy, and which causes an increase in the mortality and morbidity. In the present meta-analysis, there were 6 studies included for fasting blood sugar with 508 participants, 8 studies for glycated hemoglobin which had 599 participants. There were patients with type 2 diabetes who were randomized to follow either a high fiber diet or a controlled diet. Post the intervention, it was shown that there was a significant reduction there was a reduction in both the fasting blood sugar and postprandial blood sugar in both the groups, which is the high fiber and control. Both of them have shown a reduction. However, the reduction in the high fiber diet group was significantly more as compared to the control diet. And there was also a dietary fiber group shows that the reduction in glycated hemoglobin was much more when compared to the control group. Coming to the adverse events, these are adverse events which were reported, but then these are not much significant for a study because we have already seen that the glycemic control and short chain fatty acids have positively shown results on the dietary fiber group. However, just to emphasize that there were no adverse events reported, and the galacto-oligosaccharides were also well tolerated. And however, there was no much significant difference between both the groups, the intervention and the non-intervention control group. These results overall show that there was a significant improvement in glycated hemoglobin, the short chain fatty acids, and the abundance of bifidobacterium in the dietary group with the control group. So, to conclude, it says that the dietary fiber significantly improves with a p-value of less than 0.05 the abundance of bifidobacterium, short chain fatty acids, and glycated hemoglobin. Thank you.
Hello. Yeah. Thank you, Suleiman. So we'll go with the discussion. So before we jump, next one, before we jump into the discussion, I'll just highlight the key result part. So the dietary fiber significantly improved the fasting glucose, the HOMA-IR, acetic acid, propionic acid, the butyric acid, and also the adverse events in the patients with the type 2 diabetes. When the groups were compared, the dietary fiber group and the placebo group, when it is compared, there was a significant difference or the significant improvement we have seen in the glycated hemoglobin, but there was no significant differences was found in terms of this fasting blood glucose, the HOMA-IR, and the adverse events. So what the mechanism or because of the short chain fatty acid, the increase of the short chain fatty acids produced by the dietary fiber that has led to enhance this GLP-1, the production, and which has contributed to the improvement of this glycated hemoglobin. So next slide. So we all know that already what is short chain fatty acid, and also most of the colleagues have been covered that one. So it is a key role. It plays a key role in the health and the diseases and regulate the gut homeostasis, right? And if any deficiencies, so that if you see the deficiencies of these short chain fatty acids, again it is involved in the pathogenesis of these inflammatory bowel diseases or the cancers or some cardiometabolic disorders. So what actually these short chain fatty acids, they are produced by the friendly bacteria, the fermentation of these fibers, that is non-digestible fibers in our gut. So it's actually the main source of the energy or the nutrition for the cells, which is present in the colon. And we can say that the most common short chain fatty acids already in the introduction part, it has covered like butyrate, that is, it accounts for 60%, where propionate, it accounts for 20%, and acetate is also 20%. So once these are used as energy by the cell lining of our gut, and the rest of it, they're moved to the gut wall and entered into the blood and perform some of the health benefits. So we call it as a super organisms. So what like the human gastrointestinal, it has a complex and dynamic population of these microorganisms, right? So the gut microbiota, again, it is going exert a influence, the marked influence on these hosts during the homeostasis as well as in the disease condition. So what's the gut microbiota? In the slide, it is the gut microbiota. It constitutes the complex community that interacts with each other and with the host, and it modulate the beneficial effects on the health. So the gut microbiota, it's mainly encompasses the four main phyla, that is actinobacteria. We can see in the figure, which is already discussed. But the dominant microbial phyla accounts for 98%, in which the actinobacteria gives about 1 to 13%, and Bacteroidetes is 9 to 42%. The Firmicutes is 30 to 52%. So we can see here Firmicutes, what all the bacteria cover: Firmicutes, Bacteroidetes, Actinobacteria, and Proteobacteria. Next. So as we have already, even the systematic review also, it talks that they have the many physiological health benefits by maintaining this gut health. So again, it influences on many factors like genetic, age, physical activity, and also sometimes the antibiotics. Also, the major is the diet diversity. So if you see someone who is having the consuming the animal protein, their GI tract has dominated with the bacterioid species. So the person who is associated with the plant-based diets, they're having the species like a Prevotella. So accordingly, it is goes on varies. So here in the systematic review and the meta-analysis that have shown a significant improvements in the glycated hemoglobin, again, the total short chain fatty acids and the relative abundance, we can see here the abundance of bifidobacterium in the dietary fiber group. So even the similar results were found by the Hongton and their co-workers, the team. So there are many metagenomic studies are also revealed that the association between these two dominant phyla, that is the firmicutes and the bacteroidetes ratio, that is relatively stable in the healthy, whereas any disturbances happen with these ratios, that is firmicute and bacteroidetes ratio, that can lead to the development of metabolic disorders. Again, these ratios, it's going to change the age as the infancy, it's going to changes in the middle age. In the middle age adult, it's about 10.9 of ratios. Again, it's going to change when in the elderly persons. So the major alteration, yes, previous only. The major alterations in the diabetic, it include a significant lower abundance of these firmicutes. So usually in the healthy person, what happens? The firmicutes in the gut microbiota, it will varies between 11% to 95%, where in case of this diabetic patients, it will be a lower abundance of these firmicutes. And we can see the increased enrichment of these bacteroidetes. Yes. So in the figure on the right side, we can see the healthy intestine that with the bacterial species, microbial this thing. So the intestinal mucosa, it consists of, we know that epithelial cells will be the immune cells, and these helps in their secretions, which helps in the nutrient absorption and also protect from the external factors with their tight junction. And also the gut microbiota, that is going to participate in that, like regulating the integrity these and also the function of this intestinal barrier. So in the left side, we can see that once this gut microbiota disturbs, or we can say the gut microbial dysbiosis happens, it's going to develop the diabetic and its complications. Okay. So usually this gut epithelium is an efficient barrier that prevents the absorption of this liposaccharides, that is LPS. These LPS, they are produced usually by the gram negative bacteria, that is proteobacteria. They have, that is because they are made up of the outer layer membrane is made up of the lipopolysaccharides. So due to some lifestyle changes or the dietary alterations, so this is going to increase, that is the LPS is going to increase, and that enters into the bloodstream. That way, in terms that the leaky gut also happens. So that results in an increase in the plasma levels of these LPS. So where it is termed as a metabolic endotoxemia. Finally, it is going to lead to the inflammations and the several pro-inflammatory cytokines again in the adipocytes and all, which is contribute to the insulin resistance. And finally, it develops to be type 2 diabetes and its complications. Yes, next. So we know that the complex, we can see here the complex interaction, or we can see the communication. So mechanism that happens between the gut bacteria and the host body. Particularly if we see in the diabetes mellitus, these gut microbiota derived metabolites, no, they're having an influence, the signaling pathway, it is going to affect, they have their target cells as well. The glucose metabolism target cells, that is the hepatic cells, adipose cells, skeletal cells. So again, it is going to affect the glucose metabolism, insulin sensitivity, inflammation and all. So already most of the short chain fatty acids have been covered. So but here, what is say that it is a signal molecules which activate, we can see in the figure, okay, that signal molecules which activate the downstream of these D protein coupled receptors and also the olfactory receptors. that is again which helps in the insulin and the leptin secretions, and also it going to modulate the inflammatory this thing, and also regulate the blood pressures. So if we come to LPS, it is already discussed in the previous slide. So if we come to the next is trimethylamine, which is derived from the dietary choline, L-carnitine, and phosphatidyl-choline by the fermentation of certain microbiota species. So that is going to elevate the plasma levels of this TMAO level. That has been shown to increase again the risk of these cardiovascular diseases, non-alcoholic fatty liver, and also the diabetes mellitus. So even the enzyme or the TMAO pathway, that activation, it like a key negative effect on the glucose metabolism maintenance. So if we come to the aromatic amino acids, it consists of tryptophan, phenylalanine, and the tyrosine. So this could be generated into the again indole and p-cresol. So by the catalytic effect of these tyrosine phenylase, so in the gut microbiota, it is going to ultimately sulfated into the indoxyl sulfate and precresol sulfate by the hepatic cells. So that we call it as, we can say it as also known as what is that, uremic toxins. So that is, again, it is associated with the thrombosis or the kidney diseases, even the diabetic nephropathy as well. So this is what we can say that the gut has a major role in the diabetes. Look, yes, next. So with the following, like the most evidences, they have shown that the available on this microbial activity, they are again linked with the beneficial effect of this gut microbiota against type 2 diabetes. So some of the mechanisms that is summarized here, that is it maintenance of the integrity of this intestinal barrier, the reduction in the bacteria translocation, and consequently the systemic inflammations is reduced, and the production of the short chain fatty acids. So again, which influence on the metabolic pathways. So to get these benefits, so again, one need to focus on their diet or the nutrient balance. So as we know that the nutrition shape the gut microbiota, and it accounts over 20%. Again, it going to vary from human to human, that person to person. So we can see here this figure that shows the effect of the macronutrients on the gut microbiota and the type 2 diabetes mellitus. So we can there is different diets also it impact that macronutrient, micronutrient. We can see that how it is effect on the gut microbiota and the type 2 diabetes. So that we can this can be the important approaches to prevent and control the diabetes. So we see that complex carbohydrate have the the main in nutritional therapy in case of the management of this type 2 diabetes. Even the ADA, that American Dietetic Association, that has a diabetic association, that guidelines included the importance of the diets which is having the like the complex carbohydrate, so that it reduces the glycated hemoglobin. Helps in improving the glycated hemoglobin. Sorry. So if we come to the dietary fiber, so previous one, I'll just summarize it. Okay. If we come to the dietary fiber, again, these enzymatically, this is not enzymatically, we know that not degraded. Again, it is fermented in the large intestine. These dietary fibers, so like we can see in the figure, they have the positive correlation with the bacteria, which I've seen that is arrow mark with the upside, and some of the negative correlations have seen with the bacteria. The bacteroids, the streptococcus and all. So again, with this, it's going to like the overall abundance of these bacteroidetes is increased, and thereby improving these firmicutes to the bacteroidetes ratios. Again, it's elevate the short chain fatty acids, improve the this glycated hemoglobin levels, insulin sensitivity is going to improve. So if we come to the starches, again, these starches they are classified accordingly degree of these enzymatic hydrolysis, like rapid digestible, slow digestible, resistant starches, where here we focus on the resistant starches. Again, we have the 5 types of resistant starches. RS1, that is where we found in a whole grains. The RS2 is a negated potato starches, the green banana starch, or the RS3 is what the amylose or the retrograded starches. And the RS4 is the chemically modified starches, and the RS5 is the amylose lipid complex. So these starches, again, the foods such as the potato, rice, the breakfast, usually they contains around less than 2.5% of the resistant starches. On the other hand, the foods like boiled legumes, peas, and also the like the cooked and cooled starchy foods, no, they contain higher amount of this resistant starches, that is 5 to 15%. So when it is compared to the Western diet, it has a minimal content of the resistant starches. So again, we can see there is an increase in the bifidobacterium, parabacteroidetes and all. Again, it helps in like enhancing the GLP secretions, elevated short chain fatty acids, which helps in overcome this type 2 diabetes. So if we come to the proteins, like different types of proteins, again, it has effects on the insulin signaling. For example, like animal protein that going to promote the insulin resistant and is associated with the increased risk of diabetes. On the other hand, the plant protein, again, it promote the insulin sensitivity and have the improved control, that is glycemic control in the patients with a type 2 diabetes. Like, if we come to the lean meats, it is going to benefit the gut bacterial changes. We can see that increase of few bacterial species and decrease of few bacterial species, but they do, but they have the neutral effect on the glycated hemoglobin, though they do not reduce this postprandial glucose secretion. But in case of this red meats, generally it is going to contribute to the harmful changes. Again, it's going to promote or elevate these negative metabolites, okay, microbiota metabolites, including the TMAO or the branched chain amino acids. Again, it's going to decrease the short chain fatty acids and increase the risk of type 2 diabetes. So if we come to the fat, so most of the studies consistently, they have shown that the diets which are high in the fat, that it increases the risk of type 2 diabetes. So through the impaired glucose tolerance and also the binding of insulin to its receptors, or in resulting in this altered glucose transport and all. So again, it depends on the type and the quality of the lipids which we consume. So if we speak like more specifically, like replacing the short chain fatty acids with the PUFA, that is omega-3 and omega-6 PUFAs, so was associated with the 35% reduction in the or decrease in the risk of type 2 diabetes we can see. And also, if we replace these trans fatty acids with the PUFA, again, it is correlated with the 40% reduction in the risk of this type 2 diabetes. So again, these trans fats also promote the harmful changes we can see with a negatively affecting this leptin and insulin resistance. Again, finally, it leads to the type 2 diabetes. Next. So since our dietary habits, it has a mixture of all these macronutrients, nutrient amounts, we are continuously or like delivered to our gut ecosystem. So here we can see the different types of the diets are there, how it is going how it is going to affect on the gut microbiota and the mucous layer. So we can see here in the figure, the bacteria species variations are indicated in a rectangular frames. So the arrows pointing up and down, that is respectively, that indicate the increase or decrease of those bacterial abundance. So each color is different. They have mentioned like the rectangular frames, no, that is one phylum, that is the yellow for actinobacteria, green for firmicutes, red for proteobacteria, and blue for bacteroids, purple for a verum microbia. So if we come to the first, the gluten-free diet, so usually it is the effective treatment for the celiac diseases. So if this is studied in the healthy individuals, again for the 1 month, most of the studies have shown that so this gluten-free diet followed for 1 month, which showed that decrease in the beneficial gut bacteria like bifidobacterium, lactobacillus, and also increase in the harmful bacteria like Escherichia coli, E. coli. So despite the benefits of these celiac disease patients, but the long-term adherence to these gluten-free diet, again, it leads to the imbalance of the gut microbiota. So if we come to the ketogenic diet, so it is like a high fat, very low carb, and which is usually in the drug resistant epilepsy and the GLUT1 deficiency syndromes. So they also, we can see here, there are like disturbances in these microbial species. So that is like influencing the production of certain metabolites, like that they are going to cross even the blood-brain barrier, as well as, again, it going to affect the brain function. So if we come to the low FODMAP diet, so that is effective in managing these IBS, that is irritable bowel syndrome and all. So, but they can lead to the reduction, again, a reduction in the beneficial gut bacteria and also the overall bacterial abundance, because we can see at the total bacterial abundance has decreased. So due to the decreased intake of the prebiotics, we can say. So once if the supplementations of with this the probiotics or the prebiotics, that again, it helps in mitigating these negative effects, and we can restore the beneficial bacterial levels. So even if you come to the Western diet, again, it is like rich in the fat, the animal protein, refined sugars. Significantly, it is going to alter the complete gut microbiota compositions and leading to the dysbiosis, which again, which leads to the metabolic diseases, the inflammations, obesity, these things. If we come to the Mediterranean diet, which is focused majorly or the more on like predominant plant-based one, which have the healthy fats and low animal protein intake, that significantly benefits the gut microbiota diversity. We can see here the microbiota diversity as well the stability has increased. So the studies that integrate that indicates who are all adhere to the Mediterranean diet, so with the higher levels of these beneficial bacteria and also the increased production of these short chain fatty acids, we can see, and overall improvements in the gut health. So we can see that the nutrition is important in determining the complete makeup of this gut microbiota. So once switching to the predominant plant-based diet, that may help in increasing the diversity and also the stability of these gut microbiota, which is helps in again health promoting bacteria, which is what are the health promoting bacteria in the gut. So further in the further slides, we'll see that particularly pertaining to the dietary fiber. So what are how it is benefited in case of the diabetic patients. Over to you, ma'am. Thank you.
Thank you, Preetham. So I'll quickly summarize because we are already running out of time. So I'll just quickly summarize the remaining part of the discussion. If we quickly talk about the disrupted gut-liver axis in diabetes. So what happens when an individual starts consuming more unhealthy diet, predominantly if they consume meat-based diet, so it's going to increase and decrease the short-chain fatty acid producing bacteria, leading to gut dysbiosis. And ultimately, that is going to impact the pancreatic beta cell function, as well as leading to the increased carbohydrate metabolism, leading to further steatosis and liver damage. So the other way, this particular gut dysbiosis can also lead to the leakage of bacteria and increase in the LPS into the bloodstream, lead to the inflammatory response as well. So it's going to be like a cyclical chain that happens. Now, my colleague has already mentioned about this. So when there is gut microbial dysbiosis, increase in LPS, metabolic endotoxemia, leading to inflammation, insulin resistance, and type 2 diabetes and its complications. So in the current review, mostly they found out that galacto-oligosaccharides, which are a type of soluble fiber, they can actually ameliorate the dysbiosis in patients suffering with type 2 diabetes. So by consuming the foods that are rich in soluble fiber or the galacto-oligosaccharides, there can be an abundance of bifidobacteria, and we can see the diversity in the intestinal microbiota as well. So which can lead to a significant changes in a lot of biochemical parameters. And most importantly, these when the food rich in dietary fiber is consumed, it is actually going to lead in the increase in the production of good gut bacteria or the friendly gut bacteria, which are going to lead to the increased production of SCFA or the short chain fatty acids, which can lead to the increased GLP-1 peptide, which is going to increase the insulin secretion as well as improve the insulin sensitivity. So in addition to that, these friendly bacteria has also been reported to increase the glycogen synthesis, decrease the expression of hepatic gluconeogenesis, improve the translocation of blood transporters, and promote the glucose uptake as well. So as already mentioned, galacto-oligosaccharides are the soluble fibers, the non-digestible one. So it is also a prebiotic, which also helps in improving the integrity of the gut wall. As a result, it can help in suppressing the intestinal allergic inflammatory responses as well. So these are some of the classification, the examples of the types of dietary fiber and their respective food sources. So the galacto-oligosaccharides falls under this particular category of soluble fibers. So again, this is a beautiful representation of how galacto-oligosaccharides are going to lead to the production of short-chain fatty acids. Now, if you look at the galacto-oligosaccharides, they are going to lead to the production of SCFAs, which are going to lead to multiple benefits, like they serve as the source of energy, they inhibit the potential pathogens by antimicrobial action, they enhance the immune defenses of the host as well, immunomodulation can occur, and they improve the morphology of the gut mucosa by increasing the mucin production as well. If you can remember in the initial slides where Dr. Dharani has mentioned, when someone consumes unhealthy type of diet, the mucin production decreases, but in contrast, when we start consuming the good plant-based healthy diet, the mucin production can also be increased. And again, it inhibits the growth of the colonic cancer cells, and it also helps in enhancing the mineral absorption in the large intestine as well. And galacto-oligosaccharides also have a direct effect by interacting with epithelial cells in the gut wall, leading to an increased integrity of the gut. And also it reduces the risk of developing the inflammatory bowel disease and the risk of colon cancer as well. So these are the beautiful images that that represent whatever we have discussed till now. And one more benefit of this dietary fiber is that they also have this laxative effect, which can again add to the bulk. They are going to lead to stimulate the bacterial fermentation in the colon, and they also increase the bacterial mass, which in turn again increase the fecal bulk, and they also going to increase the water content of the fetus, and as a result they're going to stimulate the peristalsis in the colon as well. So one laxative effect can also be seen. And one more beautiful benefit of this dietary fiber is that when the food rich in dietary fiber is consumed, it's actually technically low caloric food, and it can also increase the satiety. As a result, our caloric consumption also can be controlled, which can lead to caloric surplus and weight loss. When weight loss occurs, automatically it's going to improve the insulin sensitivity or reduce the insulin resistance, and finally improvement in the glycemic control can also be seen. So here we can see the weight loss ranging from 5 to 10% has been reported to improve the glycated hemoglobin, and the obesity related metabolic risk factors were also reduced pertaining to this particular study. And a number of studies have also demonstrated that consumption of a soluble viscous fiber supplement can also improve the glycemic control, including the glycated HbA1c with the type 2 diabetes. So this is again a beautiful representation of how dietary fiber intake can overall help in reducing the body weight through the hormonal effect, intestinal effect, and intrinsic effect. If you look at the hormonal way, it is going to reduce the glucose and insulin release. Most importantly, when the food rich in dietary fiber is consumed, it's going to reduce the transit time. I mean, it's going to delay the gastric emptying, so that it also helps in better satiety level as well, and a slow entry of the glucose into the blood as well, controlling the blood glucose spikes as well. In those ways, it's going to actually help in better glycemic control as well. So this is also explained beautifully, the hormonal effects, the intrinsic effects, and as well as the colonic effects. So finally, which are going to help in reducing the body weight and improving the insulin sensitivity, and finally leading to the better glycemic control. And as I've already mentioned, the physiochemical properties of dietary fiber are significant in influencing the functioning of the gastrointestinal, including the digestion, nutrient bioaccessibility, microbial fermentation, glycemic control. So these properties have a significant effect on the physiology of digestion and gut function, including the nutrient bioaccessibility. They are going to delay the rate of gastric emptying, the transit time will be delayed, the inhibition of flow, the mixing efficiency of digester, and the effects of gut microbiota, and finally glycemic control can be achieved. So if you look at a major mechanism that has been identified in the control of blood glucose in type 2 diabetic patients is the dietary fiber in physically encapsulating or entrapping nutrients, thus slowing down the rate of digestion of plant tissues and helping us to reduce the rise in blood glucose levels, controlling the blood sugar spikes. So this is what the main mechanisms when dietary fiber-rich foods are consumed. There is going to be delay in gastric emptying. There can be a modification of release of digestion-related gut hormones. There can be a delay of amylolysis, breakdown of the starch, and the delay of sugar diffusion and absorption, which can all help in improving the postprandial glycemic control. Again, reduce the risk of type 2 diabetes or bringing a better management of the type 2 diabetes. And all this depends upon some of the factors like the polysaccharide composition, the physical state of the fiber source as well, and also whether the fiber has been processed or unprocessed, and the presence of other variables in food such as lipids. All these can again influence the gut function. So if someone who is consuming 10 grams of dietary fiber per day for 8 weeks in one of the specific study, it has been noted there is a significant reduction in HbA1c, and there is a significant reduction in fasting blood sugar, fasting insulin, HOMA-IR as well. So not only those benefits, dietary fibers are also proven to reduce the fasting plasma cholesterol levels as well, leading to further reduction in the cardiovascular risk as well. And most importantly, one more characteristic of this dietary fiber is predominantly they belong to the low GI category, which can again help in controlling the blood sugar spikes. So the limitation of this review is, although 9 studies were included, finally, the studies include the meta-analysis for SCFS and gut microbiota were no more than 3 and 2 studies, respectively, which could limit the wider application of the findings. Hence, more studies are required in this particular area of research. So if we come to the conclusion, this systematic review and meta-analysis have demonstrated that dietary fiber, inclusion of dietary fiber can significantly improve the relative abundance of the good or the friendly gut bacteria, which can lead to the increase in the total short-chain fatty acids and also leading to the improvement in the glycated hemoglobin. So if we come to a conclusion, like in a very simple way, if you look at the takeaway points, how dietary fiber can be beneficial. Now, when we start including more amount of plant-based foods or including the plant-based diet, it can, which is rich in again dietary fiber, that is going to lead to increase in short-chain fatty acids, which can lead to increase in GLP-1, that can increase in the insulin sensitivity. As a result, we can achieve the glycemic control. The other thing is, by increasing, because the dietary fiber rich foods are low in calories and they also belong to the low GI category, they can have an increase in satiety, which also again leads to the increased scope for weight loss, which again in turn leads to the improvement in the insulin sensitivity and in the improvement in the glycemic control. One more strategy is that, but through this dietary fiber inclusion, there can be a delay in the breakdown of the starch or amylolysis, which again leads to the delay in gastric emptying, delay in the absorption of the glucose, and again improved glycemic control. The other strategy can be by inclusion of the dietary fiber rich foods, there can be increase in good gut bacterial concentration, which are going to again reduce the inflammation, decrease in insulin resistance, and improve glycemic control. The other strategy can be by including the foods that are rich in galacto-oligosaccharides, dietary fiber rich foods. They again increase in short-chain fatty acids, which again improves basically the gut integrity, and that can help in reducing the gut inflammation and the cancer risk as well. One more benefit is that this particular dietary fiber rich foods can again help in reducing the cholesterol levels and reducing the cardiovascular risk. So these are the various benefits of inclusion of the plant-based diets rich in dietary fiber, which can improve through various mechanisms. It's going to lead to the improvement in the glycemic control, as well as it is also going to improve the gut health, as well as it is also going to improve the cardiovascular health as well. So these are some of the pointers that we can take away. So this is a beautiful picture that again represents the same what I have summarized. So thank you so much, everyone. So this is it for today's presentation. If you have any queries, we would love to take it. And also, please do let us know how you felt about today's session. Was it insightful or informative? Please do let us know so that we can improve ourselves in the coming sessions as well. And the next month, we'll be coming up with another interesting topic of how the plant-based diet can impact the diabetic retinopathy. Thank you. Thank you so much, Dr. Sharmi. All right. So it's already past 8:30. Thank you so much, everyone. We'll be meeting you all again in one more interesting session in the next month. Till then, take care. Bye-bye. Good night.